A nucleus as a cell organelle was first described by Robert Brown. Robert Brown is the person who described nucleus as early in 1831. Later, the material of the nucleus stained by the basic dyes was given the name chromatin. Nucleus was stained means see here, this was DNA. Here oxygen was negatively charged, and here nitrogen was positively charged. So when the dye was applied to the nucleus, this dye will look like this, and it will has nitrogen as charged particle. This is dye. Okay, it will be in blue color originally. When this was applied to DNA, this oxygen and the nitrogen of the dye both will form electrostatic bond, and the dye will fix at the DNA. So DNA will be stained blue color because dye was blue color already. When it formed with when it forms a bond with oxygen, it appears as blue as a whole. When it was stained, the name was given chromatin to it. The interphase nucleus, when the cell is not dividing, has an extended and elaborate nucleoprotein fibers called chromatin. See, this was chromatin. See, this was DNA. This was nucleoprotein. Nucleoprotein means nucleus and proteins are combined together. What are proteins? These are proteins. Proteins are nothing but they are amino acids. Amino acids will look like this. Group of amino acids we call protein. That's it. When protein is nothing but they will look like this, with a functional group and nitrogen present in it. Will look like this. Nuclear matrix. See, basically thread is this thread is this one. This thread is chromatin. And see, Here the whole is nucleus, whole the spherical thing is nucleus. It was known as nuclear matrix. Inside it, these are known as nuclear matrix. And more spherical body known as nucleoli. This is spherical and it was known as nucleoli. Electron microscopy has revealed that the nuclear envelope, which consists of two parallel membranes with a space between 10 to 15 nanometers. Means this one here outer membrane, this one, and the inner membrane and outer membrane. In between the space, this space is known as perinuclear space. This space is known as perinuclear space. Okay. Forms a barrier between the materials present inside and the nucleus. So this was inside, this was outside. So it will be the barrier. Barrier between the material present inside the nucleus and that of the cytoplasm. So the outer membrane usually remains continuous with the endoplasmic reticulum. So this was the outer membrane. It will be continued as endoplasmic reticulum. This was endoplasmic reticulum. This was the neighboring organelle, and ribosomes are present on it. Ribosome means RNA. And ribosomes are present in it. At a number of places, the nuclear envelope is interrupted by minute pores. Then this one, the nuclear membrane, this was the pore. This was the nuclear pore. Here in the diagram it will be visible like this, nuclear pore. See here it was the nuclear pore. It was the nuclear pore. From this, the materials from inside will come out, and from the materials from outside will go in. This was the cytoplasm. These nuclear pores are the passages through which movement of RNA and proteins takes place in both directions between the nucleus and cytoplasm. Anything from the nucleus should come outside will come through this nuclear pore, and anything should grow from cytoplasm to inside the nucleus will go through the same pore. Normally there is one nucleus per cell, variation in the number of nuclear frequent. So generally nucleus will be only one. This whole one will be one per cell. In many cases, two or three or n number of nuclei will be present. This number of are frequently observed. Example: erythrocytes of many mammals and sieve tube of vascular plants. In RBC, red blood cell of a human, nucleus will be absent, and the sieve tube of mature cells even lack nucleus. Vascular plants means the food pipes of plants transport water through pipes. So if nucleus was present here, it will be a problem for the water to pass through across the plant from roots to stem or roots to leaf. So this nucleus will be degenerated in vascular plants. The nuclear matrix of the nucleoplasm contains nucleolus and chromatin. So nucleus has nucleoplasm. This was nucleoplasm. Inside contains, this one was nucleolus. This one was nucleolus. Here chromatin material will be present. The content of nucleolus is continuous with the nucleoplasm, as it is not a membrane-bound structure. This one has no membrane, just it was a thickly stained visible for us. But so this material will be continuously present with it because it has no membrane. It is a site for ribosomal RNA synthesis. Larger and more number of nuclei present in cells actively carrying a protein. See this one, which was not membrane bounded, means it has no membrane, but to show it has no membrane, we had pictured it as membrane. Inside it only ribosomal RNA will be present. Ribosomal RNA will look like this. This one was present inside the nucleolus. Inside the nucleolus, single stranded RNA, this will be present. This will be get modified. This will be get into a particular design, and we call it as ribosome. These are manufactured here. These are manufactured and modified here. This ribosomal RNA was manufactured and modified here, and some of the proteins which will bind to ribosomal RNA to fit it as a design. So ribosomal RNA and proteins will be present inside the nucleolus. So ribosomal RNA is a large and more number nuclei are present in actively carrying out proteins. So which shall have more protein synthesis? This nucleolus will be larger. You may call that the interphase nucleus has a loose and distinct network of nucleofiber chromatin. So interphase, when the cell was not dividing, the DNA will be loosely packed on a network of nucleoprotein fibers called chromatin. So these was the proteins, histone proteins. Two 4 pairs of histone proteins, and DNA will wrap this protein. So we call it as nucleoprotein fiber called chromatin. But during different stages of cell division, cell shows structured chromosomes in place of the nucleus. So later this will be visible as chromosome. These thread will be coiled perfectly as this chromosome. Chromatin contains DNA and some basic proteins called histones, and some non-histone proteins, and also RNA. So as we discussed, these were the histone proteins. These were the DNA, and non-histone proteins means like which transfer this DNA's transfer the DNA's information to mRNA. So some proteins are required to transfer this information, to proceed this information as mRNA. So that they are non-histone proteins. And RNA, mRNA is RNA, or from this tRNA will also be produced. So in the place of RNA mentioned in the text, tRNA also tRNA also a term. tRNA is also nothing but RNA coiled into T shape. This one was tRNA. Same. RNA means again, as I shown there, same. This one only. It will be when the when RNA ribosomal RNA means same RNA will form into different shape. Will form into different shape. tRNA means same RNA will form into T shape. So a single human cell approximately has two 2 meter long thread 2 meter long thread of DNA distributed among it 23 pairs of chromosomes. So see these are the starting from 1 2 3 4 5 and into 23 pairs of chromosomes. This one actually, this all was 2 meters of thread changed like this from a thread like chromatin to final shape. It was condensed, it was folded morely and finally appeared like this. So finally a chromosome thread like was changed to like this. Every chromosome essentially has a primary constriction or a centromere on the sides of which disc kinetochores are present. This was the centromere. Centromere was nothing but this was all was DNA. At the centromeres, some different sequences will be present. They are known as satellite genes. Means they are not used for morely for transcription, but when the cell division occurs, microtubules will come and attach here. So if the any DNA will be damaged or to hold the microtubules, the the centromere will the position will be holded here. Actually this will don't bind to direct centromere, it will bind to kinetochore present here. Kinetochore present at its sides. Microtubules will bind to this. Kinetochore was nothing but it was also a protein, different types of protein. What is protein? Proteins are a group of amino acids. Amino acids will be look like this, nothing but. So microtubules will attach to the kinetochore. So here at the centromere, the whatever the DNA was present, it was tightly packed here. Means the histone proteins was the DNA was wrapped tightly without gaps. So wherever the DNA was wrapped like this, we call it as heterochromatin. Here, wherever the material was loosely packed, we called it as euchromatin. We call it as euchromatin. Based on the position of the centromere, it will be classified as 4 types: metacentric, submetacentric, allocentric, telocentric. Means this this centromere, this centromere the centromere position will be changed. So at metacentric it will be here. At a submetacentric this will be some more slightly upward. At acrocentric some more slightly upward. At telocentric it will be present at here. So based on the centromere positions, the microtubules attachment of microtubules during the cell division, the position will be changed. Sometimes a few chromosomes have non-staining secondary construction at a constant location. This gives the appearance of small fragment called a satellite. So this was the chromosome, and satellite means this was the satellite means ribosomal RNA genes will be present here. From here only ribosome RNA will be produced, or tRNA will be produced. This was the site where called satellite. So actually this was a staining body only. It was a staining only. It looks as staining, but it was mentioned as non-staining because exactly at this position it looked like thread-like structure. So it was known as non-stain. So when viewed under the electron microscope, chromatin appears as beads on string. So this was the histone molecules, and this was the chromatin. Again histone molecules. This star is known as beads on string, appeared as beads on string, called nucleosome. A typical nucleosome contains This thread contains 200 base pairs of DNA wrapped two turns around the core of histone octamer. Octamer means these are 8 proteins, so it was mentioned as octamer. What are proteins? Proteins are group of amino acids. Amino acids will look like this. And a single H1 protein will be present here. So, and seals the 2 turns of DNA by binding at the point where the DNA enters and leaves the core. So here it will be sealed means these 2 nucleosomes will be fitted perfectly, as it cannot move. The DNA that continues between 2 successful nucleosomes is called as linker DNA. This one was linker DNA. This one was linker DNA. So the association between the negatively charged DNA and positively charged histones allow for the meaningful DNA packaging inside the nucleus. As these histone molecules were positively charged, DNA has phosphorus, so PO4 minus, in that oxygen was negatively charged, so charged particles will bind together, so it allows for a meaningful DNA packaging inside the nucleus. The beads on string structure and chromatin is formed to form chromatin fibers that are further recoiled into chromosomes. First one, chromatin will be present like this. This one only get some more condensed. This one was chromatin fiber, and finally it will appear as chromosome. At the starting we call it as chromatin. Actually some more tightly packed we call as chromatin fiber, some more densely packed we call it as chromosome.